and I'm going to share with you my research on genetic studies of cancer. So majorly, I'm going to talk about genetic study for prostate cancer and breast cancer on identifying risk genes and pharmacogenomic study. To start with, I'm just going to give you a background introduction for the two cancer that I've studied. Prostate cancer and breast cancer are actually the most common cancer and the second leading cause of death in males and females respectively. And genetic factors are the known risk factor for both prostate cancer and breast cancer, with a heritability estimate of 42% and 27%. Genome-wide associated studies and fine mapping approaches are the most effective approaches to identify the risk genes since 2006. So GWAS actually examined millions of genetic markers on the so-called single nucleotide polymorphism SNPs across the entire genome to identify the risk genes. This approach has been extremely successful with over 1,200 published GWAS for 201 traits until end of last year. So this slide actually shows you the risk SNPs identified for prostate cancer using GWAS and fine mapping studies. So there are 5 reported in 2007, and then 14 reported, and then 33 total by end of last year. Among all the 33 SNPs reported, the four risk loci indicated by a blue bar are actually reported by our study. Just to give you an example of one of the risk loci that identified by our study, it's on HNF1B gene at 17q12. So this discovery is reported in Nature Genetics in 2008, and we use a combined of GWAS and fine mapping approach. So now people know there are 33 risk SNPs, and we want to know what's the proportion that they can explain for the heritability. Unfortunately, they only can explain a small proportion, about 14%. So people are wondering what are the missing components of the heritability. So number one, that we need to identify more genetic variants of smaller effects. And number two, that we need to evaluate gene-gene and gene-environmental interactions. And number three, the rare variants, possibly with large effect, can be identified through sequencing analysis. And number four, the structural variant, which is poorly captured by the current GWAS, need to be further explored. So my next two projects are actually focused on the missing item 2 and item 3. So my project on gene-gene interaction, which is also an ongoing project, is supported by a DoD award. And the purpose of this study is to identify SNPs in the genome that interact with each of the 33 known SNPs to have stronger effect on prostate cancer risk. So this gives you a preliminary result for this project. So this is actually a two-way interaction between one of the 33 risk loci and the genome-wide SNPs. So this figure is called a Manhattan plot, and each color here represents one chromosome, and each dot here represents one SNP. So the higher the dots here, the more significant the SNPs are. So here you can see on this figure, the top hits actually are located in the HLA region, which is indicating a potential interaction with the SNP and the HLA region to confer stronger risk for prostate cancer risk. So we need to genotype this top hits in another 2 independent populations to confirm the results. So my next project, which is also a future project on whole genome sequencing, will be supported by a pending DoD award. And in this project, we are going to do whole genome-wide sequencing, sequence the entire genome using next generation sequencing technology for 20 prostate cancer families, including 10 European Americans and 10 African American families. The aim of the study is to identify rare and novel genetic variation responsible for aggressive form of prostate cancer. So I have talked about several studies on prostate cancer. However, I did not forget about women's disease. Now I'm going to talk about an ongoing project on breast cancer. So this project is supported by a newly funded R1 in which I collaborate with Dr. Mara Lightland from the Division of Public Health. And this is actually a pharmacogenomic study trying to evaluate the gene hormone therapy interaction and the risk of breast cancer. So hormone therapy is actually the most effective approach to relieve menopausal symptoms. The Women's Health Initiative conducted a large randomized clinical trial to evaluate health outcomes upon hormone therapy. However, it was stopped early in 2002 because the increased risk for breast cancer and cardiovascular disease were observed. So the WHO finding significantly decreased the number of HT prescriptions. However, women are still suffering from menopausal symptoms. So based on this background, we hypothesized that actually not every woman have the same susceptibility of developing breast cancer with HT use. And women who are not at increased risk of breast cancer with HT use can be identified using the genetic variants. So the aim of our study is to identify those genetic variants using a GWAS approach in women from the WHI HT trial, and then confirm the results in women from the WHI observational study. So the potential clinical impact of the study is that women who are not at increased risk for breast cancer, which HTUs can be identified using the genetic variants, and those women could be prescribed HT for relieving their menopausal symptoms without increased risk of developing breast cancer. So we just started this project in this March, and we are now preparing DNA and genotyping, and we hope to see the exciting results soon. So lastly, I want to take this chance to express my acknowledgement to many people. Without them, I wouldn't be standing here today. So first and foremost, I want to thank Dr. Jin Zhu, who is the director for Center for Cancer Genomics. He is my current boss, and he was also my PhD advisor. I want to thank him for his excellent mentorship, valuable suggestions, and encouragement during all my academic career. Thank you. And I also want to thank Dr. Eugene Blaker, who is the department head for genomics and personalized medicine, and Dr. Deborah Nice, who was my previous section head, for all their support in my grant application and recommendation letter for this award. I also want to thank for all the faculty members in the Center for Human Cancer Genomics, Lily, Dr. Jing Kim, Dr. Jinji Feng, Dr. Corinne Cater, Dr. Fang Ji, Dr. Mary Vatlin, for all their support in my research. And I also want to thank for the postdoc fellow and research associate in our group, Guangfu, Jishan, and Tao, for all their support, all their help in my research. Special thanks goes to our administrative staff and genetic counselor, Aubrey, Tamara, Becky, and Jenny for all their different kind of help. I also want to thank our excellent analytical team, Scott, Sangtai, John, and Hui for their excellent ability in analyzing large-scale data. So I want to thank our graduate students, Yu and Carol. And last, I also want to thank our lab technician, Ji, Zhongrong, Shirley, Lina, Christine, and Tracy for generating such a beautiful data for us to work on. Lastly, I want to thank our collaborator, Dr. Isaacs, Dr. Harenberg, and Dr. Wingland for providing us samples for our project. And I also want to thank our Vanzo people for analyzing our data. And lastly, I want to thank everyone who came to this ceremony. Thank you.